Good afternoon. My name is Justin Overcash, and I'm a PhD student in the Department of Genetics at Texas A&M University. And today I'm going to talk to you about endonucleases and their relation to human health. What exactly are endonucleases? Endonucleases are basically just proteins which bind to double-stranded DNA and cause breaks in the backbone of the DNA, or the phosphodiester backbone. Endonucleases can be contrasted with exonucleases. If we look over here on the right, we have some double-stranded DNA. Exonucleases break the bonds between nucleotides, chewing them off one at a time, removing them from the DNA strand. Endonucleases, on the other hand, break only the phosphodiester backbone at specific locations. Endonucleases were discovered and isolated from bacteria and can be broken down into several classes. Restriction endonucleases have been around for a long time and are found in labs all around the country. They generate breaks in the backbone of the DNA at specific sequence locations and are useful because they generate overhangs, as we can see right here, which allow scientists to mix and match pieces of DNA, putting them together kind of like a puzzle. While restriction endonucleases are important, the most exciting class of endonucleases, at least at the moment, are highly specific endonucleases. What are highly specific endonucleases? Today I'm going to be talking about the CRISPR-Cas9 system, which is made up of the Cas9 endonuclease and a single-stranded guide RNA, which is shown in green. These two elements form a complex and target a highly specific sequence in the DNA, as we can see right here. Once they get to that specific sequence, they generate a break in the backbone of the sense and the antisense strand. This generates what we call a double-stranded break. The cell rushes to fix this double-stranded break, and one of several things can happen. In one instance, the cell can repair in an error-prone fashion, which leads to the generation of indels, or mutations which are made up of small insertions or deletions of nucleotide sequences. This can be utilized by experimenters to remove a gene or its functions. Alternatively, a homologous template can be used to make a perfect repair. This can be used by experimenters to insert DNA sequences, such as a fluorescent gene or other transgenes. All in all, the relatively low cost and high specificity of the CRISPR-Cas9 system makes it revolutionary. How do endonucleases relate to human health? In our lab, we use the CRISPR-Cas9 system to genetically modify mosquitoes, which are critical vectors of both viruses and parasites. Some examples of the viruses and parasites vectored by mosquitoes include dengue, which infects millions and kills thousands annually, malaria, which infects millions and kills hundreds of thousands annually, Zika, which has been a hot topic in the media recently, which causes microcephaly or head deformities in the infants of pregnant women who are infected with the virus, as well as several other viruses such as yellow fever, which kill thousands more. Genetically modifying these mosquitoes allows us to study their physiology with the hopes of finding solutions to the human health issues that they impose. Genetically modifying mosquitoes is relatively simple. To start, we take adult mosquitoes and collect their embryos, which are these tiny little things down here. Then we take a microscope and a fine needle and inject Cas9, the guide RNA, as well as any other components of the injection solution. Then we get genetically modified mosquitoes. In this upper picture here, we have mosquitoes which have been modified to express a red fluorescent protein in their eyes. And in this lower picture, we have a larval stage of the mosquito, which expresses a green fluorescence protein throughout its body. So what are the applications of genetically modified mosquitoes? There is a lot on this slide, but if you're interested in learning more, both of the papers pictured on the left are worth reading. The idea is simple though. Applications of genetically modified mosquitoes can be broken down into two categories. The first is population reduction, which revolves around introducing genetically modified mosquitoes into a population to cause population collapse. This would remove the mosquitoes and therefore their ability to transfer viruses or parasites to humans. The second is population replacement, which would introduce transgenic mosquitoes which are resistant to being infected by viruses or parasites, into the natural population, with the intent of replacing the natural population with transgenics. While both solutions have pros and cons, either would be a net positive for human health. In summary, endonucleases allow scientists to tackle complex human health issues, such as the spread of disease by mosquito vectors. With that, I would like to thank my departments, which house me, my principal investigator, Dr. Zach Edelman, and my lab, which assists me with my research. If you have any questions, feel free to email me. Hope you have a great day.